Okay, this is lecture 3 in microbiology. We're going to talk about prokaryotic and eukaryotic cells. I would remind you that prokaryotes include the two domains bacteria and archaea, and eukaryotes eukaryotes are in the domain eukarya. So all living organisms fall into three domains: bacteria, archaea, and eukarya. The eukarya include all of the other organisms other than the bacteria and the archaea. Plants, animals, protists or protozoas, fungi, etc. Okay, now a obvious characteristic of prokaryotes versus eukaryotes is that the prokaryotic organisms are much simpler. They do not contain membrane-bound organelles in the cell. They have what's called a prenucleus. They don't have a membrane-bound nucleus. They have a prenucleus. They have a area where the genetic material is located, where their DNA is located. Eukaryotic cells, eukaryotic organisms made of eukaryotic cells, do have a true nucleus, not a prenucleus, a true nucleus, which it has a membrane surrounding the DNA. Now, these are some of the other characteristics comparing prokaryotes and eukaryotes. In a prokaryotic organism, there is one chromosome, whereas eukaryotic organisms have multiple chromosomes. The chromosome, the DNA in the prokaryote, is circular, whereas in the eukaryotic organisms it is linear and there are pairs. These eukaryotic organisms are I've forgotten the word, a diploid. As I said earlier, there is no nuclear membrane around the DNA in prokaryotes, but in eukaryotes there is. The DNA is associated with a protein called histone. There are a lot of histone proteins in the DNA associated with the DNA in eukaryotes, but not prokaryotes. There are no membrane-bound organelles in prokaryotes, but there are in eukaryotes. The cell wall in prokaryotic organisms, there is peptidoglycan in the bacterial cell wall, but not in archaea. In the cell wall of those eukaryotic organisms that have a cell wall, not all do. Our cells, for example, do not have a cell wall, but fungi do and plants do have a cell wall. There are polysaccharides in the cell wall. Algae also. They all have polysaccharides in the cell wall. Plants, algae, fungi, all members of the domain eukarya. Cell division in prokaryotes is by binary fission. Everything doubles and divides, whereas in eukaryotic organisms it's a very carefully orchestrated sequence of events called mitosis. All right, so let's focus in on the prokaryotic cells. They range in shape. They're small, certainly. They're smaller than the average eukaryotic cell. They range in these sizes. You don't have to memorize them, but I would I can tell you, just to get an idea of size, a red blood cell, a human red blood cell, is 8 micrometers in diameter. So that should help you, I hope. There are basic shapes, different basic shapes. There are many, many shapes that bacteria can take. I'm going to restrict most of the discussion of prokaryotes to bacteria because there are no archaea that cause disease in humans. And most of what we're going to do in this course is, in fact, bacteria, very little about archaea. In terms of bacteria, the basic shape, there are many shapes, but the most common basic shapes are round, coccus, rod-shaped, bacillus, and spiral. An example of that is shown, well, spiral is spiral. I'll give you examples later. Although you already know that Staphylococcus aureus is round. You might know that E. coli is round, and you probably don't know it, but Treponema pallidum, which is the bacterial organism that causes syphilis, is spiral. There are, as I said, many other possible shapes that certain representative bacterial species have, and there are some unusual shapes, but most have one characteristic shape. Most are monomorphic. A few can take on different shapes through their, depending on conditions that they're growing in and through their life or through their life span. But most stay in one shape, and there are odd shapes other than the standard basic coccus, bacillus, and spiral. Now, another characteristic feature, and it's used for identification when a bacterial smear of bacteria is made on a microscope, and on a microscope slide, and the slide is examined under the microscope, then the way they're arranged can actually give a pretty good hint at what you're looking at. It can help with the identification. For example, the genus Neisseria, and as I go through the course, I'll keep referring to specific names, in other words, genus and species names of certain bacteria, because you do have to know them, and we will be learning more and more about them, and they do cause important diseases. So I'm just introducing them as I go along. So an example of a bacteria that you might see on a microscopic slide being examined would be Neisseria. The genus Neisseria has two species members that cause important diseases in humans. Neisseria gonorrhea. I'm going to leave the disease that it causes up to your imagination. And the other one is Neisseria meningitidis. And again, you should be able to figure out, I would say, using a little imagination there, what disease Neisseria meningitidis causes. Neisseria organisms typically are paired. They're double. And there's actually didn't notice it until now, there's a diagram of a pair of bacteria, and you know, if you saw this under the microscope, you would or should be suspicious that you're looking at a member of Neisseria. There are other characteristics. So this is an arrangement of more than one cell, how they typically arrange with each other as they grow. So some form pairs, other form clusters, some form long chains. Remember, I told you before that if not in a diagram, but in text, if the text is in text that I've put onto the slide, if the text is in black, that's just for your information. If it's in red, you need to know that. Okay. Now, in terms of actual structure and we're going to sort of go through the different parts of prokaryotic organisms in the next few slides. In terms of the outside, the very outside of many bacteria, there is a structure called a glycocalyx. It's outside of the cell wall. It helps the bacterial cells to attach. All bacteria need to attach in order to colonize whatever they're going to grow on. They need to attach in order to be able to stay in one place and grow into large numbers. So attachment is quite important, in the ability to attach is quite important, and it's mediated by the glycocalyx. The structure outside is called a capsule. Not all bacteria have a capsule. Some organisms are able to form a capsule and others aren't. Even if it's the same organism, some are encapsulated and some are not encapsulated. It's a trait that is coded for by the DNA. So some certain species of bacteria, and I'm thinking of one in particular that we'll talk about later, but there are examples where if you look at one species of bacteria, some members can form a capsule and others can't, and these capsules are an organized kind of glycocalyx as opposed to just simply material that's extruded to the outside and causes them to be sticky. It's a more organized structure, and it helps to prevent phagocytosis. Preventing phagocytosis would be important if it's an organism that enters into the body tissue and could cause a disease. Phagocytic cells, and you know that you have two major types of phagocytic cells in the human body, macrophages and all macrophage lineage cells, and that's one, and the other are neutrophils, also called neutrophils. The other granulocytes like basophils and eosinophils are somewhat phagocytic, but not anywhere near as phagocytic as neutrophils. The two major phagocytic populations of cells in the body are macrophages and neutrophils. And these cells phagocytose anything foreign to the body if it's insoluble particle, and will try and destroy it. So they're very important in protecting the body against bacterial invasion, and if the bacteria has a capsule that can prevent phagocytosis, then that bacteria is more pathogenic than a type that does not have the ability to form a capsule. So if the exterior extruded material of the glycocalyx is organized, it's called a capsule. If it's unorganized, it's simply a slime layer. Moving on to another structure that's found outside of the cell wall. Many, not all, bacteria have flagella. Okay, these projections shown here in red here in this diagram are flagella. They are outside of the cell wall, and they are made of chains of a protein called flagellin, and they are attached by a structure in the cell wall. I wouldn't worry about the exact shape or anything but that structure, but there is a structure that holds them into the cell wall, and they're involved in motility, movement of the bacteria. The flagella can have different kinds of arrangement, and often the arrangement of the flagella is a identifying characteristic that helps identify what species you're looking at. Some are, there's a single flagella at one end. There might be one flagella at either end, or there could be, as shown here in this example here, there may be a tuft. That's no good. No, that would be no good. Here, a tuft of flagella at one end, or they may be all over the body of the bacterial organisms shown here. You don't have to know these terms, but the fact that there are different arrangements can help identify. I can tell you that the example in C, just by looking at it, I can tell that it's a member of the genus Helicobacter. And Helicobacter has one species that causes disease in humans. It's called Helicobacter pylori, which causes can cause peptic ulcers in humans. All right, so this again shows that the flagella are embedded in the cell wall, and I remind you that the cell wall is here. This whole area here, I'll change color here. This entire thickness is the cell wall, and below that you have the cell membrane, okay, the plasma membrane or cell membrane, and of course internal to that is the cytoplasm. So the flagella are involved in movement of the cell, and they rotate. They act kind of like a little spiral propeller. And they can allow the bacteria to move towards or away from different stimuli. If it's a positive signal, then it'll move towards it. Positive signal might be something like glucose, so they might be able to carry out chemotaxis towards the higher concentration of glucose in a solution, or they might move away from it. If it's something that's nauseous or toxic, they can turn and move away. So movement is taxis. And when you look at the bacteria under the microscope in a culture, you see them with some difficulty going in all sorts of directions. Okay, some organisms, particularly spiral bacterial organisms, have an internal flagella. It's called an endoflagella or axial filament. You get that in spiral organisms. Spiral organisms are called also spirochetes, and they cause the whole organism to rotate so that it can move through the medium that it's in, the fluid, whatever fluid it's in. There is another kind of extension from the cell membrane outwards. These are called Fimbriae, and there are also structures called pili. They're very, very, very fine structures, much finer than flagella. And the Fimbriae are involved in attachment. The pili are specialized structures that allow for the transfer of DNA from one bacterial cell to another. So not all bacterial cells are able to carry out transfer. They need to have pili on their surface. They can use that to transfer DNA. So DNA can actually be transferred from one organism to another. And that's quite important because that is one mechanism through which a bacterial species that has the gene coding for a particular type of antibiotic resistance could transfer it to another organism. So if you have some bacteria in you that are non-pathogenic and have that gene in them, it is possible that if you also have an infection with a pathogen, that they could transfer the gene to that pathogen and make the pathogen antibiotic resistant to a particular antibiotic. That's an important consideration, and that does happen. Okay, so as we move inward, we go to the cell wall. Now, the cell wall is an important, very stiff structure, much, much stronger, in fact. This cell wall here is much stronger than the cell membrane and allows the the bacterial cell, to avoid osmotic lysis. So if it's in a hypertonic or hypotonic, let's go with hypotonic, that's what would cause bursting. If it's in a hypotonic solution, whereas a cell that doesn't have, like a eukaryotic cell, like a red cell, for example, that doesn't have a cell wall, if that was in there, it would lyse, but the bacteria wouldn't because it has that very strong cell wall. Remember, part of the strength of the cell wall is provided by a molecule called peptidoglycan. Gram-positive organisms have a very thick peptidoglycan layer, and gram-negatives have a thin peptidoglycan, but both gram-positive and gram-negative bacteria do have peptidoglycan in their cell wall. This is a diagram showing the structure of peptidoglycan. It's enough for you to know that it is a polymer of disaccharides. Many disaccharides strung together in a particular pattern that are linked together by polypeptides. That's why it's called peptido. Peptido referring to the polypeptide part, and glycan referring to the carbohydrate part, the disaccharides. And so it has a particular kind of structure. If you look, as I told you before, at gram-positive organisms versus gram-negative organisms, remember, different bacteria can be stained, most bacteria can be stained using the gram stain and will be either gram-positive or gram-negative. The gram-positives have a thick peptidoglycan layer, and the gram-negatives have a thin layer of peptidoglycan. Gram-positives have teichoic acid, gram-negatives do not. Gram-positives have one plasma membrane. It's quite unusual, or at least you haven't you may not know this at all, but gram-negatives have two membranes. They have an outer membrane and the inner plasma or cell membrane. So the outer membrane is outside, actually. Well, I'll show you a diagram that clearly shows that. Gram-positives and gram-negatives, these organisms have toxins typically, and the toxin in a gram-positive organism is called an exotoxin. It is secreted out by the organism, so the organism releases it. The big difference between gram-positive and different gram-positive bacterial species will have different exotoxins, all right? Toxins that they secrete will be unique to each different one. The endotoxins are found in gram-positives. Endotoxin, in fact, is not secreted, but is part of the cell wall of gram-negatives. But as the gram-negative organisms die, and bacteria, even when they're growing, a few are dying in the population, you know, a certain percentage are always dying. They're gradually releasing endotoxin as they break down. Okay, this is the shows the difference in the makeup of the gram-positive cell wall versus gram-negative cell wall. The major differences that we've already discussed include the fact that there is a much thicker layer of peptidoglycan in the gram-positive cell wall. It's shown here. I'm highlighting it, as opposed to this layer there. Now the other thing is that the gram-negative cell wall also has a outer membrane. The gram-negative cells have a outer membrane. So there's a normal plasma membrane, the usual plasma membrane here, this layer here, and then the cell wall itself is made up of a layer of peptidoglycan and an outer membrane. So there's a second phospholipid membrane. I would point out to you also that the outer membrane in gram-negative organisms includes a molecule called lipopolysaccharide. Lipopolysaccharide is part lipid and part carbohydrate, so that's why it's called lipopolysaccharide. The lipid portion is lipid A, and that's where the endotoxin that is what makes up the endotoxin. So this is the toxic part of the organism. LPS, the abbreviation for LPS, lipopolysaccharide, the abbreviation for lipopolysaccharide is LPS. Every gram-negative organism has it in its cell wall, and it's the lipid A portion of the molecule which is toxic. The other part is called an O polysaccharide. Another key distinguishing characteristic is the fact that the gram-positive cell wall has teichoic acid in it, whereas the gram-negative does not. Okay. So the gram-positive cell wall has teichoic acid. The polysaccharides that are present allow for identification of variants or subtypes, strains, so that they can be identified based on the variation that can be detected with antibodies. Antigenic variations. Different antibodies will respond to the different polysaccharides that are present, and it can be different within different strains of a particular species. Gram-negative cell wall, again, the endotoxin and lipopolysaccharide, or LPS, are synonymous, and the toxic portion of the molecule is lipid A. The polysaccharide, again, provides the ability to distinguish between variants using different antibodies that react differently to the different types. The other major difference also is the outer membrane, so that it in fact has two membranes, the inner plasma or cell membrane and an outer membrane, which forms a significant portion of the cell wall itself. And this is everything that I mentioned here is in the previous two slides is written out here. The outer membrane does protect from phagocytic cells. Well, it protects against a number of mammalian immune defense mechanisms, including phagocytic cells. Complement activation. Complement is a antibacterial set of antibacterial proteins found in the plasma. The outer membrane can also protect against certain antibiotics. Okay, so this basis of being able to distinguish gram-positive from gram-negative allows us to identify most, not all, bacteria. Most, but not all bacteria are in fact able to be stained by using the gram staining technique. The gram stain therefore allows identification of these two major groups, and is a fairly quick, easy, inexpensive proceeding or test or procedure that can be quickly done and can help determine the proper treatment. The procedure itself, in terms of addition of reagents, and you're going to be doing this in a virtual lab, the staining procedure itself of the gram stain, there are more steps than this. These are just a listing of the reagents that are added. They're added on and then washed off. The initial stain is crystal violet, and it will stain the bacterial cells, both gram-positive and gram-negative. It'll stain them a dark blue or violet. Once the crystal violet is, once the bacteria is stained with crystal violet, then the next reagent that is added is iodine. Iodine will cause the formation of crystal violet iodine crystals. All right, so these are abbreviated CVI crystals. So you get these an interaction between the iodine and the crystal violet to form these crystals inside the cells. And at this point, the cells are still all dark blue, dark violet. Then alcohol is added, which may or may not dissolve the crystal violet iodine crystals. If it's a gram-positive organism, because of the thick peptidoglycan layer, the alcohol dehydrates that layer and it becomes impossible for the crystals to dissolve and leave the bacteria. So the bacteria remain dark violet color. However, with gram-negative organisms, because the peptidoglycan layer is thin, it will be able to dissolve the outer membrane, causing the formation of holes in the outer membrane. And, dissolve the outer membrane and forming holes in the thin peptidoglycan layer underneath. And the CVI crystal violet iodine crystals are dissolved and wash out. So now the bacteria are no longer stained violet. And in order to be able to see them, they have to be counterstained. So a counterstain has to be added to make them visible to the eye under the microscope when they're prepared on the microscope. The counterstain used is called safranin, and it will stain the bacteria red. So the order of the sequence is crystal violet to turn all the cells violet color, iodine to form the crystals, alcohol to dissolve the crystals in the gram negatives, but not the gram positive, and safranin is added so that the gram negative organisms, if there are any, can be seen, and they will be seen as red. So gram positive is violet, and gram negative organisms will be red. Okay. There are a few bacteria that do not have a cell wall. One genus or one group or genus are mycoplasmas. They lack a cell wall. There is, in fact, one important pathogenic member of this genus, Mycoplasma pneumoniae, which can cause atypical pneumonia. Archaea also do not have a cell wall. They do not have peptidoglycan. If they do have, well, they could have a small cell wall, but it's quite insignificant, really. So, and they don't have peptidoglycan, as I've said before. Let's look at the plasma membrane. It's very similar to the plasma membrane in mammalian cells in that it is a phospholipid bilayer. The outer membrane also actually is a phospholipid bilayer, but the cell membrane or plasma membrane present in all bacteria is a phospholipid bilayer with membrane proteins embedded in it. Some are integral proteins that are embedded right in it. Some are actually embedded in it and go straight across. Here's an example here of bacteria that penetrates right down through. So some are transmembrane. Some of these integral proteins. Some proteins are simply associated with the inside surface of the phospholipid bilayer cell membrane. These are called peripheral proteins. You should be aware, and you should know from your previous studies, that the integral proteins, some have all different kinds of function. Some act as receptors, some act as channels, some are pores, etc. The phospholipid membrane is not fixed. It is a dynamic structure. It has some fluid properties. It is as viscous as olive oil, in fact. Proteins can move across the surface and aggregate or associate with each other under particular conditions. The phospholipids themselves can move. And that's why it's called the fluid mosaic model of the cell membrane. You can get movement across cell membranes. Cell membranes are selectively permeable. They only allow certain things across. Other things, other molecules, they will only allow across if the right signal is received and a channel or a pore opens. Some other molecules will move right across. Movement right across from areas across a membrane, from areas of high concentration to low concentration, is called simple diffusion. If a dissolved solute associates with a transporter protein in the membrane to move across, then that's called facilitative diffusion. And here you see an example of movement across membrane, and there you see transporter protein. So this is facilitated. So facilitative transport across the membrane. The movement of water. If water moves across a selectively permeable membrane, that's called osmosis. And osmosis will occur, that movement of water will occur if the concentration of a solute is higher on the inside than the outside and the solute cannot get out. So if this, and you did this lab last year using dialysis tubing, if a sausage section of dialysis tubing is selectively impermeable to a particular solute inside the fluid inside, then, and it's surrounded by water, then water molecules will move across and try to essentially the water is moving from an area of higher concentration to an area of lower concentration relative to water. On the outside here, the water is 100%, and the inside it's not, because it has some of this solvent dissolved into it. So water will move across, and the process is called osmosis. This should all be reviewed, this idea of movement across selectively permeable membranes. It's important to remember it, and that it also happens in bacterial cells, not just in mammalian eukaryotic cells. So here you see a bacterial cell in an isotonic solution, in a hypotonic solution, and in a hypertonic solution. I would remind you that in this case, unlike eukaryotic cells that only have a cell membrane, the bacteria also have a cell wall. So they're actually quite resistant to bursting, but they will burst if put into a highly hypotonic solution. And with a hypertonic solution, they undergo a process called plasmolysis, in which case the interior shrinks because of the osmosis of water moving out. Water moves out by osmosis into the hypertonic solution on the outside, and the inner part, the cytoplasm inside of this cell, therefore is reduced in volume. So you get this shrinking in of the cell membrane. You have here, I'll highlight it in black, you have a cell membrane around, this is the cell membrane or plasma membrane of this cell. It's now pulled away from the cell wall. The volume of the cytoplasm is being reduced because of the osmosis. That is called plasmolysis. It takes a lot to burst much higher, a much lower concentration of solutes, so a much lower hypotonicity for a bacterial cell to burst versus a eukaryotic cell like a red blood cell. Now membranes, cell membranes can be damaged by other types of molecules, and these are the kind of molecules actually that are used to disinfect or damage bacteria, because they do have an effect on the plasma membrane. So alcohol is a pretty good agent to cause damage to the plasma membrane or to cell wall. Detergents can also damage the bacterial plasma membrane. And some antibiotics, and the example I would think the classic example would be, for example, penicillin, will also damage bacteria because it damages the cell wall. Others can damage the actual cell membrane. Okay. We talked about transport like facilitative or simple diffusion or osmosis. None of those require any energy. There are other transport mechanisms which are active transport as opposed to passive. Active transport requires energy, and the energy is drawn from stores of ATP inside the cell. Okay, I think it's worth stopping at this point, and that will be lecture 3, part 1.